Now that was, acid production from parietal cells, and we understood why they make why they make acid. So let's try to understand now regulation of this acid secretion. And regulation is really important because without regulation, it's a big mess. So let's try to understand what are the substances that can upregulate and what are the substances that can downregulate. Yes. So factor that can upregulate or increase are first is histamine, second is gastrin, third is acetylcholine, fourth is coffee, fifth alcohol, and calcium. So these things, right, they can upregulate. So let's try to understand each of them, by their singular, ways. Let's try to understand first. Let's talk about histamine. So histamines are produced by ECL cells, right? And ECL cells are enterochromaffin like cells. Now note that these enterochromaffins are like, right? They are not the enterochromaffin cells. Enterochromaffin cells particularly make serotonin, but these are like cells because they look like that. And these are part of this gastric gland, right? We have 6 type of cells in gastric glands: surface, neck, parietal, ECL, chief, and this antral gastric gland, right? So ECL makes histamine. Now let's try to understand the story of acid production, right? So what happens here is that this histamine, right, and this ECL, right, this ECL, that it's a part of a paracrine, acid production, right? So what is paracrine? Paracrine means it's neither endocrine, it's nor exocrine, right? It's in the middle. So that's what paracrine is. So paracrine acid production. How? So histamine, it's a stimulator, and histamine diffuses with something called as parietal cells. Now we already know parietal cells make acid. So histamine can directly affect parietal cells, and they bind to something called as H2 receptors, right? And as you can think, H2 receptor are primarily found in gastric acid secretion, and they are also present in several other parts of the body. For example, they are present on the parietal cells, right? They are present on the heart, they're present on the blood vessels, right? So histamine, it directly like stimulates this H2 receptors, and they just make HCl, right? So that's histamine for you. Other thing is gastrin. So gastrin, right? The story of gastrin starts with antral gastric cells and antral gastric glands. So these are glands. It means they will have cells inside them. So these are bigger parties. These are bigger collection of glands, right? Collection of cells. So they have something called as G cells. Okay. Now the thing here is that this is a gland, so it means it has multiple cells in it. First type of cell, it has something called as mucus cells, right? And they produce mucus as well. Second is this G cell, right? They produce this something called as gastrin, right? It's a gastrin, it's an hormone, it stimulates parietal cells, right? And it promotes mucus cell growth, right? Second, other thing that they have, this part, right? This antral gastric gland has something called as D cell. Now, wherever we hear the word D, we should remember somatostatin, right? Now, this somatostatin, it's an inhibitor. It's an inhibitor of everything. Okay. Now, this somatostatin can be released from G cell. It can be released. Sorry, it can be released from D cells, right? I'm so sorry, not from G cell, right? And that's it. So this is antral gastric gland. It produces G cell. Now, gastrin is released, and gastrin, directly increases acid production. How? So just G cell, and it's generally kept under a neural inhibition because promotion of these, upregulation of G cell can lead to upregulation of acid, and it can upregulate acid mediated destruction, right? So the trigger for the release, okay, are amino acid chains, vagal stimulation, and stomach distension. So these 3 things, right? Amino acid protein, vagal stimulation, it means something is messing up with your cranial nerve number 10, and it's not acetylcholine, right? So how it works is this, that vagus nerve, right? It's called vagus because vagus means wandering, and it's a nerve that comes from your, not comes, I mean, it originates from your brain and it goes to your stomach, and then it's like diffusely spread all over your body. So that's why it's called vagus. So the vagus nerve stimulates this gastrin release from the G cell, yes, but it's not directly with acetylcholine. That's very important to understand because it works indirectly. Okay, it works via this. Okay, let's try to understand this. Acetylcholine, it's a neuroexcitator, right? It's a neurotransmitter that excites, right? And this acetylcholine, it acts on something called as enteric neurons. Okay, the neurons that are present in the this enteric plexus. Okay, so once what happens is that this once the acetylcholine acts, it releases on it acts on these neurons, and these neurons, they release something called as gastrin releasing peptide. Now this is called GRP, right? So acetylcholine releases GRP. So this gastric releasing peptide, this directly stimulates G cell, and then from G cell we get gastrin, and from gastrin we get HCl. So that's the concept about vagal stimulation. So vagal stimulation, stomach distension via stomach distension. So your stomach is like that. I'm making a stomach. So this is how your stomach looks like. Usually distension happen right here, and here are these stretch receptors. So what happens here is that once your stomach is distended, it will lead to stretch receptor getting activated, and once they are activated, we will have release of G cells, not release of gastrin via G cells. And so what happens after the story, right? So gastrin is now made, right? Gastrin goes and binds with something called a CCKB receptor. Now we have two type of receptors, CCKA and CCKB. That's very important to know. So gastrin acts on CCKB receptors. Now these CCKB receptors are present on parietal cells or something called as ECL cells, right? So they get binded together, and they they love each other, and this parietal cells get activated, and now this is a GQ. Now GQ leads to increased CAMP activities. Now CAMP activities lead to H plus K plus ATPase upregulation, and then we have more acid. And this gastrin can also bind to ECL cells that can directly release histamine, and then histamine can go and bind with parietal cells, or it can bind with H2 receptors, right? And then it will directly or indirectly will lead to release of more acid. So I hope we understand gastrin and histamine. Now let's talk about acetylcholine. So acetylcholine is a wonderful neurotransmitter, and let's study about this acetylcholine, how it can do. So acetylcholine is released in this enteric neuron system of our stomach, right? So acetylcholine comes from post-ganglionic parasympathetic fibers. So what I'm trying to say here is that this is our nerve system, right? This is our, for example, this is our brain. This part is brain. This is your spinal cord, right? Here is your stomach, right? And here is one ganglion. So what is a ganglion, by the way? Ganglion is just bunch of neuronal, what should I say? Nucleus? Not nucleus. It's just a collection of small processes. So what I'm trying to say is that this post-ganglionic parasympathetic fibers, right? Post-ganglionic means after the ganglion, and parasympathetic fiber is just this second neuron in the parasympathetic nervous system, right? And this is the one neuron that goes from the ganglion to the target organ, right? So I hope you understand this. It's like it's a two neuron chain. Okay, so we have a parasympathetic pathway, right? It's a two neuron chain. Now we have something called as preganglion, and then we have postganglion. Okay, preganglion. What is preganglion? Preganglion is just the cell body, right? The cell body is in the brainstem, and or maybe it's in the sacral spine, depending upon whatever example we are taking. So from that cell body, this is a cell body, an axon is coming out, right? For example, from the brainstem, an axon is coming out, and right at this ganglion, right, another ganglion, right? So this axon fuses with this another ganglion, and after this another axon starts. So this another one is the postganglionic. This is a preganglionic. I hope you understand this concept, right? So the neurotransmitter usually used in the post and pre is both acetylcholine, right? I'm talking about parasympathetic system here, right? Parasympathetic is just relax and eat, right? So in both sense, for pre and post, the neurotransmitter used is acetylcholine, because it's a excitatory neurotransmitter. But in a preganglionic, this preganglionic system, we have N2 receptor, that is nicotine, and in the postganglionic we have usually muscarinic, that is from M1 to M5. So that's the idea I'm talking about. So acetylcholine comes from this post-ganglionic parasympathetic fibers. It triggers something called a cephalic phase or gastric distension. So cephalic phase, what the hell is cephalic phase? So we have three type of phases in acid secretion. We have cephalic, we have this we have this gastric, and then we have intestinal, right? So we have three type of phases: cephalic, gastric, and intestinal, right? So cephalic phase is just your brain working and secreting, acid. Gastric is your stomach, your enteric neurons really thinking, and okay, oh, you have food, let's release some acid. And intestinal is that, okay, there is some food in duodenum, let's try to digest a little bit of protein. So that's the idea of intestinal phase. So this cephalic is actually controlled by your acetylcholine. Okay, so acetylcholine goes and it triggers cephalic phase, right? And it leads to acid secretion. Cool. How? Whenever we think about something tasty, something really amazing, we feel something, we are salivating and we have increased gastric secretion in our stomach. Gastric distension, for example, we eat a lot of thing and then there is a distension of our, so we have, I told you over, this is our stomach, for example, and this is duodenum sphincter, this is LES, the lower esophageal sphincter. So distension usually happen here because this is a very thin, it's not a very thick membrane. Here's a very thin membrane, here's a very thick membrane. Okay, so distension happen in the upper right corner, or whatever you want to call it. So gastric distension happens. Once the distension happens, this distension has this stretch receptors, right? Stretch receptors, they can sense the stretch. Once these receptors, they sense the stretch, the ECL, right, the enterochromaffin like cells, they produce histamine directly causes, directly diffuses on the parietal cells, and then parietal cells make HCl. And distension, again, this again, G cells, gastrin release, we know already about it. Again, this distension can cause on M3 on parietal cells. I told you on the pre-sympathetic and post-sympathetic, pre-parasympathetic and post-parasympathetic. Pre-parasympathetic works via nicotine receptors, and post-parasympathetic works from muscarinic, that is M1 to M5. Okay, so these are like just names of receptors. These are just addresses of the receptors. So parietal cells usually have M2 receptors, and acetylcholine is a postganglionic, parasympathetic fiber. It touches on these parietal cells. It's amazing how it works. And we have these M3 and then acetylcholine, and it gets secreted on this M3. M3 gets activated. Once M3 is activated, there's this potassium and not potassium, H plus and K plus ATPase upregulation of these pumps happen, and then H plus goes into this glandular lumen, and this K plus goes inside our blood vessel, and this K is being exchanged for some chloride, and then this chloride eventually goes to the lumen of the gland, and then we have HCl in the lumen of the parietal cell. So that's how it happens. Cool shit, right? Amazing. Coffee, amazing thing. Amazing. I love coffee because I want to have some gastric ulcers. That's why I want to have coffee. Okay, so coffee. So the cool thing here is that even a decaffeinated coffee can cause gastrin release. So if you think that, okay, oh, I have ulcers, okay, I will drink some decaf coffee, it's not going to give me ulcers. No, it will give you a lot of ulcers, even because the coffee inside the coffee, the caffeine inside the coffee doesn't cause HCl release. Okay, that's important. So even decaf can cause gastrin release. Now we know that gastrin can act on ECL, and ECL can cause histamine, and histamine can cause, HCl production by H plus K plus ATPase, right? And so that's one point. Another point is that even a mild stimulation can cause, vagus stimulation, right? So coffee actually is sympathetic, stimulation, right? It is a little bit, it stimulates your sympathetic chains. So it stimulates our vagus nerve. Vagus nerve again releases some acetylcholine. We know that acetylcholine can work via ECL, G cells, M3 cells on parietal, and we understand that. So this is 2 points. First, okay, why coffee suppresses hunger? I mean, these are just my notes, why I'm thinking. I mean, why does it even happen? So what happens is that caffeine does not, increase gastrin. So gastrin is the number one hormone for your, this thing called as, okay, let's talk about it. Caffeine does not increase gastrin, but other compound on caffeine increase the gastrin. Okay, so gastrin, we remember gastrin comes from G cells, right? And G cells are present in enteral cells, right? The last cells, the sixth ones, right? So G cells make gastrin. Gastrin is negatively inhibited by these neural inhibition. Once this trigger of what? Amino acids, trigger of amino acid, trigger of distension, and trigger one more trigger. What was that trigger? Vagal stimulation, yes. So we have these three trigger. They can produce a gastrin, right? So here what I'm trying to say is that coffee does not increase gastrin, but the other compounds that are found in the coffee can increase gastrin, right? So gastrin is not the main mediator of hunger. That's cool shit right there. Hunger comes from something called as ghrelin. And what happened here is this: ghrelin works via oxyntic cells of the stomach. I will tell you about this. What happens here is this: oxyntic cells of the stomach, they directly communicate with hypothalamus, right? And this is how you get hungry. We'll talk about it. It's an amazing concept. So it doesn't, it really doesn't suppress anything. This is just one thing that, oh, I thought that it increases gastrin. Okay. Second, it increases acid. How? Via a negative feedback loop. Okay. Now how? This increased acid goes to duodenum and you feel satiety because your stomach feels that, oh, not stomach, duodenum, right? So what happens is that once we have increased acid, secretion due to coffee, and then this increased acid goes to our duodenum, right, which is right there, and our duodenum have some receptors for these acid. It can sense, and once it senses that, oh, you have some acid coming from the stomach, it feels that, oh, he must have eaten something. Okay, once it feels that you have some acid, or it feels that, okay, you must have eaten something, what happens is that you have this negative feedback loop and you feel that, oh, you're not hungry anymore. So that's how one way coffee works. Other way coffee works is that it triggers sympathetic system, right? It activates it, and it increases a little bit of adrenaline, right? It increases a little bit of cortisol, and then you feel alert and not hungry, right? Because who want to feel hungry when they are stressed, right? Cortisol is a stress hormone, you know that, I hope. Alcohol, cool shit, right? Alcohol, it irritates the mucus, and it stimulates vagus, right? And it leads to acid secretion on the surface, right? So we understand this. Irritates the mucosal surface cells. Okay, these surfaces, they make mucus. Okay, calcium, cool thing. Calcium is a G cell stimulator. Okay, and we know that G cell make gastrin, and gastrin can directly affect parietal cells, right? So that's one way to think about it. Cool. Let's talk about these, okay, sources. So histamine comes from ECL. We understand gastrin comes from we know this already. Acts on H2 receptor, histamine, H2, right? So gastrin acts on cholecystine kinin B, right? CCKB, which are present on the parietal cells. Okay. Now note that these CCKB receptor can be present differently on a lot of organs. So that's one concept to understand. And acetylcholine works via M3. We know that muscarini's family, they stay under post parasympathetic fibers, right? So right, we understand this. What else we're trying to understand? So these are the substances that can upregulate acid. Okay, the substances that can decrease the acid are excess acid via negative feedback loop. It's amazing how it works. Somatostatin, that's one of the universal inhibitor, right? Via D cells that are present in the enteral gastric gland. Okay, so not enteral, they're not really present inside this gland. They're present right here. So somatostatin can stop acid. Prostaglandin, it's amazing. So prostaglandin, especially E2 is one of the main prostaglandin for pain and fever. So E2 and I2, what happened here is that this E2, it goes and it decreases the CAMP levels, right? And it decreases this CAMP level because it works via GI, right? That is okay, let me try to explain you a little better. So let's try to understand what is prostaglandin first, right? Because if we don't understand prostaglandin, we will not be able to understand this CAMP shit. So what happened here is that what are prostaglandins? Prostaglandins, PGs, are locally acting these protein mediators, right? These are protein. So I'm trying to understand protein. It means they must have some sort of second messenger. That's very important. So prostaglandin, they locally act, right? And they are okay. I was so wrong. They are not okay. They are not protein. They are fat, okay, because they come from arachidonic acid, AA, and it's a fat. Okay, they are lipid mediator. They are not protein mediators. I just got confused. I'm so sorry. So E2 is a fat mediator, right? It's an inflammatory mediator. It's made up of fat, it's made up of lipid, and it's made up of arachidonic acid. So if we don't have arachidonic acid from our diet, we will not be able to make good prostaglandins. It's funny because in Russian, prosta means just. So I'm just thinking about it right now. So they're a part of this, leukotriene thromboxane family. And how, these prostaglandin is made is that they come from arachidonic acid. Now, arachidonic acid is is like a membrane phospholipid, right? And then arachidonics can make this something called as PGG2 or PGH2, right? So we have this enzyme called like cyclooxygenase. It's called COX. A lot of people have it. Okay, it's a good joke. So a lot of people have this COX, right? Cyclooxygenase, COX, and it's called as prostaglandin endoperoxide synthase. And it's a synthase. It means it makes two shit. So there are two type of COX. We have COX1, we have COX2. Okay, so COX1 is like a housekeeping service, and COX2 is usually famous for inflammation. So what happens is this, that these two, COX1, COX2, they make this prostaglandin. Okay, so that's very important. Okay, so now we understand what's this prostaglandin, right? So the role of PG, that is prostaglandin E2 in stomach, is that it's a protective mechanism, right? So why did God even thought, okay, let's give you this E2 for your protection? Because a lot of time what happens is that we have a lot of acid secretion in our stomach, and it can cause a lot of inflammation on these cells, right? So this prostaglandin is one of the part how negative inhibition, negative feedback loop for reducing your acid in stomach works. So this PGE2, it binds. And how, okay, let's talk about the whole story. So this PGE2 is a protective mechanism. God gave you this because inflammation can cause a lot of it's great to have inflammation, because God was like, okay, let's give you some PGE2 so that you can reduce the acid secretion. So on parietal cells, it decreases acid, right? So we have something called as GI coupled receptors, right? GI means G protein inhibition. I stands for inhibition usually. So I'm thinking, so PGE2 binds with these receptors on these parietal cells. Now I don't remember the name of these receptors, right? But these receptors are present on the membrane, and now this membrane it activates something called as GI protein, that is glycoprotein inhibition, right? So this GI, it stops something called as adenylate cyclase. AC, I usually call it AC. So once AC goes down, we have less activity of CAMP, and it usually leads to less protein kinase A activity, and then it usually leads to H plus that's proton and potassium pumps getting really really negatively just negative. I don't know how to say this. They get really messed up. That's what I'm trying to say. So we have less acid production. So PGE2 leads to decreased level of CAMP activity in parietal cells, and then we have low levels of H plus K plus ATPase, right? What about I2? I2 usually leads to increase in mucosal flow. So that was, okay. What about how about paracetamols? Do you know about NSAIDs like paracetamol, right? So we know about these NSAIDs, and these NSAIDs they block this something called as COX. We already, and COX make PGE2, right? And once we can stop this PGE2, so NSAID blocks this COX, so we have low levels of prostaglandin, especially PGI2 and E2, both are low level now. So imagine we don't have brakes on acid. So I mean, now what's going to happen? Less mucosal protection due to less E2, more acid damage due to less E2, and we will have something called as gastric ulcer. So that's how you will have gastric ulcers from persistent intake of paracetamol. Okay, what about other drugs called misoprostol, right? So misoprostol, it's actually it's how it works. It mimics prostaglandin. That's it. So it's like you can take this analog of prostaglandin that is present in this external world, and you can buy it in a shop, and then you buy it, and then you have less acid production, and then you have more mucus bicarbonate production. And usually we give this misoprostol, right, in prevention for acid for NSAID induced ulcers. Okay, so that's what I'm trying to say. Okay, cholecystokinin, amazing story. We must understand that this is one of the best moment. I know. So one of the best concept. So we have this, this hormone called cholecystokinin. Now, the trigger for cholecystokinin is fatty acid, amino acids, right, in small intestine. That is very important because if we have this fatty acid and amino acid, it means you have eaten something. When you have eaten something really good, not just something very high in carb and nothing in fatty acid, nothing in amino acid, you will not have good levels of cholecystokinin. Usually the theme in GIT is that most of the things, most of the hormones that are regulated, that are released, are due to some sort of fatty acid presence or presence of amino acid or some sort of distension of the stomach. Usually carbs are not one of the biggest activators of hormones in GIT. So fatty acid and amino acid, they lead to release of cholecystokinin when they are present in the small intestine. Once they are present in the small intestine, the CCK is released. Now CCK has two choices. It can go, it does two things, not choice. It increases the bile and pancreatic flow, and then we know that bile is like a detergent, and then it cleans the fat, and it makes these little fat droplets called emulsification, right? So fat is digested because of bile and pancreatic enzymes. Now what happens after this? The CCK, it acts on same CCK B cells. Now cool thing is that here CCK B cells, they decrease acid, but if we talk about here's something called as gastrin. Now, when gastrin act on CCK B cells, on parietal cells, it increases acid. How? It's like a paradox, but it's not really a paradox. We'll talk about it how. So just be with me for this story. So CCK goes and binds with CCK B cells, and it decreases the acid release. And CCK B cells, right? Now let's talk about CCKB because it's really interesting. So we have these three things, right? So these three things, what are these three things? We have increased acid, we have decreased acid via CCKB, and then we have this, okay, just we have two things. I was wrong. So CCKB, it binds with something called as GQ, right? GQ, and GQ is just for calcium, remember that, right? So we have something called as, we have three type of this G protein receptors. We have GI, we have GS, and we have GQ. And GI is for inhibition, GS for stimulation, GQ is for increased levels of calciums. Okay, usually I'm not saying it's always like that, it is usually like that. So GQ is receptor found on pancreas, gallbladder, D cells, and neuronal cells. Neurons of enteric system, neurons of gastric system, right? Not okay. So what happened here is that we know that GQ leads to increased level of calcium intracellularly, and usually calcium is associated with exocytosis of exocytosis of hormones. So increased level of intracellular calcium leads to pancreas releasing their enzymes, gallbladder releasing their enzymes. Well, not their enzyme, gallbladder just releases, concentrated bile. That's the story of gallbladder. But D cells make somatostatin. So increased level of calcium via GQ, stimulation leads to stimulation of somatostatin, right? Somatostatin is a universal inhibitor, so it stops acid. So that's one story. Neurons, they make acetylcholine here in enteric system, and we know that enteric system, this acetylcholine can positively and negatively affect our acid production, right? So it is amazing how it works, right? So This is how one way the CCKB can act, right, via GQ. Another way via this, this whole shit can happen is that, let's talk. So another way is that this CCKB, right? This CCKB, we already talked about this cholecystokinin and this something called as gastrin can bind with CCKB, right? So CCKB recognizes gastrin and CCKB both, but it's a big but. How? So eye cells, right? Eye cells that are present in the duodenum and ileum makes CCK bind with D cells, and then we have somatostatin release, right? So what I'm here trying to go is that the location of CCK is very different, right? Now CCKB receptor is present in a lot of places. What I'm trying to say, right? Here I've written that why body uses the same receptor, CCKB receptor, for increasing the acid and for decreasing acid. I mean, it's quite inefficient if you think about it, but it's not really inefficient, because I'll tell you how. Because both gastrin and CCK recognize this CCKB, and both gastrin and CCK are related peptide. They come from one family, so they have similar structure. So both of them, they can bind with CCKB. Now I was thinking, oh, maybe there's some sort of competition between them. Usually whoever is more in number will win, then bind with this. But it's not usually the story. The story is not like this. What happens is that these gastrin and CCK, they can bind to same CCKA and CCKB usually. And what happened here is that the CCKB leads to stomach function, and CCKA leads to intestinal functions, right? But also it can lead to a negative feedback of CCKB. So imagine if CCKA is getting affected, so it will stop CCKB function. If B is, it will not stop CCKA. So that's the story of this CCKB and CCKA, right? There are two receptors. So here, what the whole story is happening, here what's really happening here is that the CCKB is present at different places, and at different places it will act differently. Okay, at CC, if it's on parietal cells, and if it is related with GS or GQ. For example, here it's related to GQ. It means it increases the calcium levels in the intracellularly, and it will lead to exocytosis of these things, right? Okay, so depending upon the location, CCK receptors can act differently. That's good concept. I think good concept to understand. Okay, for example, eye cells, right, in duodenum and in ileum, they make CCK, and this CCK binds with D cells, and D cells make somatostatin. So that's very good concept as well. So that was right. Let's talk about secretin. So okay, let's talk about secretin right now. So secretin, cool name by the way, secretary, like a secretary, right? Secretutka. So secretin, it comes from S cell, yeah, not so smart naming. So S cell, they are present in duodenum mucosa. That's very important point. So the whole thing here is that the acid chyme or acid that comes from stomach, because you have just drunk, you were so hungry, you thought, oh, let's drink a cup of coffee. So you just had your cup of coffee, and now a lot of acid is getting secreted in your stomach, and stomach is like, oh my gosh, what should I do with this acid? So acid will be thrown inside this duodenum. When they open this sphincter, duodenal sphincter, it will open, and then acid will flow inside this early duodenum. And at this place, we have this acid sensing receptors. They will sense acid is here. So S cell that is present in the duodenal mucosa will be like, oh, we have so much acid, we need to neutralize it. So what happened later is that this S cell will release secretin. Now this acid which is present in the duodenum lead to inhibition of G cell. We know G cell make gastrin, and gastrin leads to more acid, right? Okay, so G cell stopped, no acid. It leads to inhibition of parietal. We know parietal is the primary source of acid. Okay, no acid now. What about stimulation? This secretin, right? It's a secretary for what? It's a secretary for pancreas. So it goes to pancreas and say, help us. We have so much acid in duodenum. Please do something. So pancreas is like, oh, okay, let me just contract myself and pinch myself, and just change myself a little bit, and just poop out a little bit of bicarb inside the duodenum. So it just poops out a little bit of duodenum inside, a little bit of this bicarb inside your duodenum, and then this acid is getting neutralized. So that's the story of secretin right there. What about calcitonin? Calcitonin means calcium that is toning. Tone means to, to tone, to tight it down, to really tight it up. So it reduces the levels of serum calcium levels, right? So calcitonin, it comes from parafollicular cells of thyroid, right? We have, so this is a thyroid. I'm making a thyroid gland. We have these cells, right? They make T3, T4. And then we have these cells, right, around these thyroid. We call them parafollicular cells. Follicular cells make T3, T4. Parafollicular cells make this this calcitonin. Okay, so calcitonin made, right, they come from parafollicular thyroid, right? The trigger for these calcitonin release is high levels of calcium, high level of calcium in our blood. We know that calcium is a signaling molecule. It can lead to, a lot of mess in our cells. We don't want a lot of calcium outside. So what will happen is this calcitonin will be released. Once it is released, this calcium will decrease the level of this calcitonin will result in decreasing calcium serum levels, and it will act on inhibiting G cells. Now it inhibits G cells. We know that G cells are gastrin. Gastrin leads to more acid. So no gastrin, no acid. It leads to not inhibition, down regulation of parietal cell activities. So calcitonin works on two cells, parietal, and there's something called as this G cells. Okay, so now we know calcitonin, secretin, cholecystokinin, prostaglandin, somatostatin, negative inhibition loop. We know that. We know histamine, we know gastrin, we know acetylcholine, we know coffee, we know alcohol, we know calcium, right? So that was whole regulation of gastric acid secretion.